Hello, good evening. You have raised your hand. Is there any question?
Hello, ma'am. Good evening.
Good evening. Yeah.
Ma'am, at what time the class will start?
I'm just waiting 2-3 minutes if anybody joins, otherwise we'll start.
Okay, ma'am. Okay.
Let's start today's session. Very good evening. I myself is Tanishka. I am a PhD student from University of Hyderabad and we'll discuss the week 11 content from the NPTEL course Enzyme Science and Technology. So, yeah. So, week 11 content was the application of enzyme. Okay. About the application both in food industry and as well as in medical field. Okay. So, in food industry, as we can say that enzymes are extensively used for many different purposes, like what we get from the farms and everything. Those are the raw materials or raw foods which we get. Okay, and that final product which we get in the market, that is done by some engineering using the enzymes, like the color, the texture, the appearance of the fruit or any other eating product. Okay, in brewing also enzymes are used, also for the food modification, and there are several other things for dairy products, fermentation and all the processes takes place, cheese formation, curd formation. So everywhere enzymes are being used. So those are the categorized in food industry. Other than that, in the medical field also extensively enzymes are used for the diagnosis purpose, for like they'll check in the blood what are the enzymes that are present in the high level or the low level, and accordingly they'll correlate that thing to the disease. So whether it is high, whether it is indicating any disease or something, so like that. So about those application, the week 11 content was divided into two: application of enzymes in food industry and application of enzyme in the medical field. Using that only, we are going to solve few questions today. We'll see the first question: which of the enzyme is used for the production of cheese? So out of these four options, can you answer like which is used for the production of cheese? You are there in the call. Please try to answer if you remember anything from the videos or
Yes,
something.
ma'am.
Yeah,
Which
please
of the
try to
enzyme
answer. Yeah.
for the production of cheese? Ma'am, I think alkaline protease.
Uh, no, it is not alkaline protease.
Okay, ma'am. So, ma'am, what is the answer?
It is rennet.
Okay, ma'am.
Okay. Do you
For
remember from where rennet is extracted?
no, ma'am.
It is obtained from the fourth stomach of the calves. Okay.
Okay, ma'am.
Yeah,
Stomach of the carbs.
the cow. Okay. Fourth stomach of the calves. It is extracted, and this rennet enzyme is used for the production of cheese. Okay. And let
Okay.
if we'll talk about the other, so papain is
Carbs.
used for the tenderizing purpose of the meat. Like we get in our home the meat delivered, so that is
Yes.
very tender. So that process is usually done by the papain. Okay,
Okay.
papain is extracted from the source of papaya. Okay, papaya is the source for the papain. So it does nothing but breaking of the peptide bond. So in the muscle or in the meat, whatever peptide bonds are present, it will break that so that it can become tender. So that is the function of papain.
Yes, ma'am.
And yeah, pancreatic enzymes. It is you can say like it used in the leather industry as well, like for the softening of leather.
Yes, ma'am.
The alkaline, these are, I'll tell you what are those purpose. As far as I can remember, for the removal of hair from the hide, something like that. Okay.
Okay, ma'am.
Yeah. So we'll see which of the following is used to tenderize the meat. So we just now discussed. So the answer will
Ma'am,
be hmm.
ma'am, pepine.
Yeah, Pepin. And why Pepin?
Ma'am, I don't know exactly, but
No, no. Pepin, what does it do? It is, do you know the function, like basic function of peptide? Cleavage of peptide bonds, right?
yes, ma'am.
Cleaving peptide bonds. So by cleaving those peptide bond, because what we get in meat, that is like muscle, okay, muscle mass, where protein is stored. So it breaks those bonds and the peptides or the amino acid chain. So that in that way, it making it tender. Okay.
Okay, ma'am.
Yeah, so the packed meat which we order to our home that generally get processed by this pepin, and the source of the pepin is papaya.
Okay, ma'am.
Okay.
Yes, ma'am.
We'll see the next question. Which of the enzyme is used for the removal of starch from beer and fruit juices? Yeah, nowadays
Ma'am,
it is like very much like cut of starch. So for that, people are using many industries are using different enzymes. Yeah.
yes, ma'am.
Which one
Ma'am, I
is?
think amylase, ma'am. I think.
Yeah, amylase.
Yes, ma'am.
Yeah. Can you tell the function of other two? Titinase and glucose oxidase.
No, ma'am.
Okay.
Ma'am, cheating is. I think, ma'am, first one we are discussing, ma'am, for the first one.
Mm-hmm.
But ma'am, really, matlab, exact don't know
Mm-hmm.
exact, ma'am.
Yeah, no problem. It's okay. And glucose oxidase, the name suggests it oxidizes the glucose.
Yes, ma'am.
Okay. Glucose oxidation, it gives rise to gluconic acid and hydrogen peroxide, some oxidizing agent it is. So in that way, wherever it is needed, glucose oxidase is used for the industrial purpose. Okay.
Okay, okay.
Yeah. And we'll see the next one. Yeah. Which of the
And
enzyme has an anti-stalling effect?
ma'am, fourth one.
Yeah. Fourth one. Anything else you know about this enzyme?
No, ma'am.
Yeah, you remember.
Yes, ma'am.
Okay. Good. Good. Okay. Yeah, it is. The answer is correct. Maltogenic amylase is the enzyme.
Yes, ma'am.
Yeah.
Which of these increase the production of alpha? Ma'am, ma'am, I don't know the answer of this. Can you explain, ma'am?
Yeah, yeah, yeah. So for the production of this amylase, there are whole procedure and many factors that affect it positively, some affect it negatively. So there, if it is asked that which of these increases the production of alpha amylase by increasing the permeability of the cell membrane. So twin
Yes.
20 and cholic acid both can do the same function independently, not together, but independently. Twin 20 and cholic acid can enhance the production of the alpha amylase due to the increase in permeability of the cell membrane.
Okay, ma'am.
Okay, so first and two
Yes.
both are correct. The third option both is the correct.
Okay.
So the next, yeah. So it is again about alpha amylase only. It is a metalloenzyme which contains which of the
Ma'am, I think, ma'am, sodium. Sodium. No, sorry, ma'am, zinc.
no, it is not zinc.
Ma'am, but I am not sure about this.
Okay. It contains sodium. Sorry, it contains calcium. Calcium is the
Yes.
correct answer. Okay,
Okay,
it is a
ma'am.
metalloenzyme that contains calcium. Okay, and at least one calcium it contains. It number varies up to 10 as well. But in this enzyme structure, at least one calcium they contain in this family.
Ma'am, and what is the function of this, ma'am? Calcium.
Uh, you can say just these are the like for the basic we read now metals
Yes, ma'am.
are there in the enzyme structure, so that's why they are called as metalloenzymes. Maybe this is helping in forming some kind of interaction with the substrate of this particular enzyme.
Okay, ma'am.
Yeah, so calcium is the answer.
Okay, ma'am.
Yeah, soya sauce can be produced by the action of which enzyme? Think.
Ma'am, uh,
Mm,
amylase.
mm, no, it's
Uh,
Chitinase.
chitinase.
it's proteases.
Oh, okay, ma'am.
The
Proteases.
proteases. Yeah. This week, um, yeah. Let's tell me. Let me go through the reason.
Soya sauce have a protein substance.
Sorry.
So
Yeah, yeah,
protein destruction process.
yeah. Protein destruction
So it's
process.
called as proteinase.
Yeah, that I know. But it helps in better emulsification, increases the emulsifying effect, and that is what proteases are used for the soya sauce production.
Okay, ma'am.
Okay, thank you.
Okay. Yeah. Which of the enzyme is used to dissolve blood clots?
Blood clots, ma'am. Lysosome.
Time.
No, no. Lysozyme is the like breaking of cell walls. So that's not the function.
Asparginase. Asparginase.
No, so the answer is urokinases.
Okay.
Urokinase. Okay.
Yeah.
Used to
It,
dissolve
yeah,
blood
it is a
clots.
serine protease and it is prepared from the human urine and it helps to dissolve the blood clots.
Okay, ma'am.
Yeah.
Okay.
Urokinase, na, ma'am?
Yeah.
Okay, ma'am.
Urokinase will be the answer for this one. Yeah. And asparaginase is used for the cancer treatment. Okay.
Cancer. Okay, ma'am.
Yeah.
For my medicinal purpose, ma'am, or for my
Yeah,
chemotherapy.
medicine. Uh, that I cannot recall right now. I cannot tell you exactly, but this has been used in the medical field. That I can tell
Yes,
is
ma'am.
the cancer treatment.
Okay, ma'am.
Yeah. For the medicinal purpose, I guess I don't think it is involved in the chemotherapy somewhere.
Yes, ma'am.
Yeah. Okay. So we'll see the next. Yeah, it's the same question. Which of the following enzyme is used for the treatment of the
Yes,
cancer?
madam. Asparaginase.
Yeah,
Asparaginase. Now only
asparaginase.
we discuss.
Yeah, asparaginase. Yeah. And yeah, pectinase can be derived from the sources such as
Such as
Pectinase,
all of
citrus fruit.
yeah,
Citrus fruit.
yeah. It
citrus.
can derive from the citrus fruits.
Citrus.
Yeah, it is citrus fruit, like including oranges and tomatoes and berries. All those are the source for the pectinase enzymes. Okay.
Okay.
Yes, thank you.
Let's see. Yeah, which of the following is not an application of pectinase? So where all the pectinases are used.
Ma'am. Third one.
Yeah, recycling of plastic. It doesn't have any application there, but it is used for the production. The main purpose is the wine and juice processing of the pectinase,
Yes.
and also it has been used for the production of coffee, chocolate, juice clarification. But
Yes.
recycling of plastic is not a application of pectinase.
Okay, ma'am.
Yeah, for the amylase production. Yeah. So for the producing amylase, many things are like for any enzyme production, many things are given. Salts are given, medium are given, nitrogen sources are being provided. In nitrogen sources, tryptone, peptone, everything is being included. So, but few ions also play important roles. So for the amylase production, which of the particular ion reduces the production by 50%?
Madam, I think zinc plus.
No, sir, it is actually magnesium. Yeah.
Okay.
Yeah, magnesium reduces the production,
Okay.
but on the other hand, calcium increases the production.
Right, sir.
Yeah,
Okay.
yeah, calcium increases the production. And other than that, all the like pH, temperature, and at what temperature one strain is producing the protein, those all things are very, very important for all the kind of enzyme production. Yeah. Today I didn't made many questions. Whatever questions were there, it was like very kind of direct questions, not
Yes.
any analytical ones. So if you have other than that anything you want to discuss, it is open discussion. Last week content also, it was nobody there, so we couldn't discuss anything. If you have any doubt, you can ask me. Okay. Have you gone through this notes, medical field, food industry, all the applications and everything?
No, ma'am.
No, not yet. Have you solved the question provided by sir, like during the assignment?
Yes, ma'am.
Yeah, those you have solved. Okay.
Yes.
These are the also similar kind of questions I've prepared, which can be helpful for you. Okay.
Okay, ma'am.
Other than that, any last week contents thing you want to ask anything or anything other than that?
Ma'am, ma'am, you can tell anything about the course, ma'am. Ma'am, because this is my first class now.
Oh, you haven't attended.
Yes, ma'am.
Okay. Okay, so in the Enzyme Science and Technology, it's a 12-week course. In all the 12 weeks, there are different topics. We started with the very first, what are the enzymes, what are the classes of the enzyme. So if you can name the classes of the enzyme, must you must be knowing.
Ma'am, classes of enzymes,
Yeah, enzymes classes. You must be knowing oxidoreductase, lyse.
ligase, ma'am, or
Yeah.
isomerase.
Hmm. Yeah, to those classes we have
Yes,
discussed till now. Other than that, we have studied the structure of the enzymes. What are those enzymes made of? So, enzymes are generally made of protein, and some are also RNA in nature. Okay, those are called as ribozymes.
yes, ma'am.
Yeah, and other than that, though mostly the enzymes are in proteinaceous nature, so we have studied till now about the primary structure of the protein, secondary structure, tertiary structure, quaternary structure, their interaction. How will you know if a protein is in a dimer, in a tetramer, and what are the techniques you can do? Okay, and if you
Yes,
want to produce any enzyme, like suppose you have one particular, like suppose amylase, you want to produce amylase you in your laboratory. Okay, so what are the information that you need to have in your mind? like like suppose you have the gene sequence of the amylase. Okay, and you have
yes,
an then what will you do? What will be a molecular biology approach for the production of this enzyme? Can you tell anything? Okay, what is your background? From biochemistry or like any other field?
ma'am,
Life science?
botany.
Botany. Okay.
Yes, ma'am.
Okay, so for botany also, have you heard about the cloning technique?
Yes, ma'am.
Yeah. So there also, like suppose I give you one gene, okay, that
Yes, ma'am.
you need to clone. Okay, so what is your approach? And if I want to ask, can I ask you, like, do you know the vectors that we use for the cloning purpose?
Yes, ma'am.
Yeah, can you tell me?
Ma'am, I think for ma'am, ma'am, bacteria are artificial chromosomes,
Mm-hmm.
and ma'am, mammalian artificial chromosomes.
Okay, yeah, artificial chromosomes, but generally for the cloning purpose, for the overexpression purpose, we are using a circular plasmid. Okay,
Yes, ma'am.
the
Yes,
circular
ma'am.
plasmid generally. Uh, okay, so circular plasmid we use. Okay, and
Yes,
if I ask you the property of plasmid, so there are like origin of replication. Okay,
yes.
and it also contains one site called MCS. You know the full form of MCS?
No, ma'am.
It is multiple cloning sites, where different restriction endonuclease sites are situated. Okay, so in the same this whole MCS region, there will be different restriction endonuclease. Suppose BAMH1, NCO1, NOT1, SAL1, many. Okay, and using any of the restriction endonuclease, you can insert your gene in between this vector or in between this plasmid.
Okay, ma'am.
Okay, so that is a basic thing you need to have to produce the protein from the gene. Okay, you have to
Yes,
prepare your gene separately, put restriction site, cut this plasmid with those same restriction sites. Suppose you have amplified your gene. Okay,
yes,
you
ma'am.
have put restriction site of like BAMH1, you have put restriction site of SAL1 at another end. Okay, you have digested it. Now you have the, with the sticky ends, you have your insert prepared. Okay, so it will go and ligate to the vector only if the vector is digested with the same enzymes. Okay, so then it will be ligated to the vector and you'll have a new construct with the vector with your gene of interest. Okay,
Okay.
and now this whole construct, if you put inside the bacteria, you provide induction and you try to overexpress it, the protein can be produced inside a bacteria. Okay, so this recombinant
Yes.
protein can be produced inside a bacteria that you can purify. Okay,
Okay.
yeah, and then we have also discussed in this class about the techniques for the purification or for the identification of the protein. Like if you have many different proteins in your sample, like if you have a mixture of protein, how will you separate which protein is there? How will you identify? What are the different techniques to identify the structure of the proteins? Those things also
Yes.
we have discussed. Okay, so
Okay,
everything in
ma'am.
one class, it will be difficult to tell for me, but yeah, like from the top I can tell, like X-ray crystallography can be used for to determine the crystal structure. Okay, where their X-ray are used, it will give a reading. That reading can be plotted and the structure can be denoted from there. Okay, those are the X-ray
Yes.
structure. And then if you want to know what are the exact atoms and molecules are involved, you can go for the NMR. Okay,
Yes,
NMR
ma'am.
technique can be used, and then so that NMR can exact tell you the readings in the forms of atoms. Like if you have put your sample through a carbon NMR, it will give you a position, how many carbons are there, at which position are there. Using that information, you can plot what is the structure coming
Yes,
out.
ma'am.
So those are the NMR structure. Other than that, we have also studied the enzyme and substrate interaction. Okay, what are
Yes.
the method, how they are interacting, and about the kinetics as well. So for the enzymology, you must have heard about the Km and Vmax, right?
Yes, ma'am.
Yeah. So what are those Km and Vmax? Can you tell me what is Km?
Ma'am, KM is, ma'am, I think, ma'am, sub substrate concentration, ma'am.
Mm-hmm. Substrate concentration. What specific? Any kind of specificity about it?
Ma'am, this is helping for half of velocity, ma'am.
Yeah. Yeah. Sir, you were also telling something. Hello. Someone else was also telling. Okay. Yeah, so Km is the constant substrate concentration at which
Yes,
the rate of reaction is half of the maximum velocity. Okay,
yes, ma'am.
that is the substrate concentration. So using that also we have, yeah. So using that Km and Vmax, all those properties also, we have solved many questions in the previous classes. Okay,
Yes,
those all the slides and everything is available on the Google Drive. The link must have been shared with you. Everything is there. Okay,
okay,
you
ma'am.
can go through and try to solve them. Okay, other than that, we have also studied about the inhibitors. Okay, so
Okay, ma'am.
inhibitors of enzymes. So there are two types of inhibitor. One is
Yes,
irreversible,
yes, ma'am.
another one is reversible inhibitor.
Okay, ma'am.
Okay, so as the
Yes,
name
ma'am.
suggests, you can like find out what is the difference. Okay, so if irreversible inhibitors is bind to the enzyme, so it completely destroys the functional site or the activity of the enzyme. Okay,
Okay, ma'am.
yeah, so irreversible inhibitor, so it completely destroys the functional group, the activity cannot be restored. Okay, and
Okay,
in case of reversible inhibitor, the activity can be restored. Okay, the functional group isn't completely destroyed or eliminated. Okay, so in the reversible only we have three types of inhibitor: competitive, non-competitive, and uncompetitive. Okay, so due
yes,
to
ma'am.
presence of those inhibitors, the Kmax Km, Vmax, all these properties sometimes changes also. Like in presence of competitive inhibitor, the Km of the enzyme increases. Okay.
Okay,
So
ma'am.
for a good enzyme or for a good activity of enzyme, the lower the Km, the better the enzyme. Because the lower the Km, the better the affinity of the enzyme towards its substrate. Okay. So
Yes.
if any competitive inhibitor is present, it increases the Km because the competitive inhibitor binds to the same site, same active site of the enzyme where the substrate binds. So if the inhibitor is binding, substrate won't be able to bind. You need more and more of substrate to remove the inhibitor, and so that substrate can occupy that site. So in that case, the Vmax remains the same, but to reach the Vmax or to reach the half of the Vmax, more substrate concentration is needed. So for that purpose, the Km increases in the competitive inhibition. Okay. for
Okay.
for other non-competitive one, where the inhibitor doesn't bind to the active site, it binds to any other site other than the active site. So in that case, the Km remains the same because there is no competition between the inhibitor and substrate for the same site. So the Km will remain the same, but the Vmax and everything will change or decrease. So these are the things that happens in the category of
Yes.
reversible inhibition. So regarding those also, few numericals also we have solved in the last class. Okay.
Yes, ma'am.
Yeah. So yeah. So now we are like just studying this application of the enzyme in the food industry and the medical industry.
Yes, ma'am.
Yeah. So anything you want to ask other than that?
Ma'am. Which is the main application, ma'am? Which is used, ma'am, of enzyme?
These two both are the applications. Okay, it's not like enzyme mainly applied into one field in the food industry and the medical. Both field the enzymes is like equally used. Okay, like in the industry also, there are drugs that needs to be made, like the medicines that we are taking. Okay, that also needs enzyme to be prepared. Okay, so those things are also there. Industry level also, enzymes are highly exploited for the production of drugs that can be used to cure. Okay,
Okay,
and
ma'am.
those and that's what, like, it is nothing but a chemical reaction, right, from a substrate getting the product. So suppose your final drug or the medicine is the final product, you have one substrate that can provide that product, but the intermediate is the enzyme that carry that reaction. Okay,
Yes,
so
ma'am.
there are in chemical catalyst as well. Okay, that can carry that
Okay.
reaction, but those chemicals are very hazardous in nature as well. Okay,
Okay, ma'am.
so replacing them, we can use the this biocatalyst, or you can say enzymes. Okay, for a green reaction to take place. Okay, there
Okay,
will
ma'am.
be like less hazardous for the nature, and you can like genetically or engineer also the enzyme. Like suppose there are one enzyme is there, it is having some active site, and the substrate is binding to the active site, and if you try to do some manipulation into the active site, you replace some another amino acid. Okay, like if in the active site serine is there, you try to replace it with the
Yes.
glycine or any other glutamate, positively charged, negatively charged, any amino acid. Try to see what are the changes that is happening. Okay, if the substrate affinity is getting substrate enzyme affinity is getting increased upon the mutation, so that is a beneficiary step. Okay, that is very
Okay, ma'am.
beneficial. It can, if it can increase the production of the drug, and a safer form can be made. Like for any drug, generally there are many drugs that have many different forms. Like if any drug has made of a carbon source, if carbon is chiral in that particular drug, it has two forms, R, S, like that. Okay, It is
Yes, ma'am.
having two enantiomers. So sometimes it happens that one enantiomer is very like life saving, okay, but the other
Okay.
enantiomer could be very harmful for the body. That can show the side effect, like we take the medicines, so sometimes we have some side effects, right? So those side effects generally caused by the other enantiomer, so there are need to reduce the other harmful enantiomer production. So for that also enzyme engineering is being done nowadays extensively to get a very like one very beneficial kind of enantiomer and lessen the production of the other kind of enantiomer. Okay,
Okay, ma'am.
so those things are being used, okay, for the industry purpose. These things are going on, okay, and also in the diagnosis also these enzymes are highly exploited, okay, for the diagnostic purpose. And in the food industry, as we have all discussed, many small steps modification of the food in the brewing industry, and in the this what can say, to cut the starch amylase is being used, right?
Yes, ma'am.
So all these uh, um foods needs to be processed by enzymes, so there also enzyme uses highly happening,
Okay,
okay.
ma'am.
So in the both the fields, food industry and the medical field, both places it has been used extensively.
Okay, ma'am.
Yeah. Anything else anybody want to ask?
No, ma'am.
Anyone else? You have registered for the exam?
Yes, ma'am.
Okay.
I asked a general question,
Sorry?
madam.
Yeah?
May I ask one general question?
Yeah, please.
You are interested to do the MCQ or NPTEL allotted to you to do the mentor section, madam?
Um,
Uh,
I
Anyone do it or NPTEL only allotted the person to do the MCQ for problem solving session, madam?
like I have been allotted to this. I applied and I have got this
Applied.
MCQ solving. Yeah,
Okay.
yeah.
Very good, madam. Very good session. I appreciate you, madam.
Thank you. Thank you. Today, even though there was nothing much to discuss because it was like very direct questions, so yeah, maybe another class
Okay.
we can discuss. We can have extensive discussion.
Applied, madam, apply only through mail.
Yeah, through mail.
Uh,
Yeah.
Through mail.
Mm.
Application form is there in mail.
Actually, I'm a PMRF fellow at my university,
Very
so I can apply. So like that,
okay, madam.
through that channel, I applied.
Okay, madam. Okay. Thank you, madam. Thank you very much for a kind information. It's an informative session, madam. Thank you very much.
Thank you. So if we have like no questions, we can conclude the session as well. Anybody want to ask or add something? Anything I should add to my slide while discussing? Or if you want to like discuss
Hello.
any particular topic, maybe
हाँ
in
मैं
the
अगर
next
लेता हूँ क्या?
presentation, yeah, I can add. Yeah, yes, please.
Ma'am, you can tell anything about ma'am, which is important for exam.
That I cannot tell. That though, for this practice, for your practice of exam only, we are doing the questions. So you can like go through the all the live interaction PPT which we have discussed, all the sir's questions. So regarding that only it will come. So
Ma'am, online videos are too long, ma'am.
you put the speed on 2x.
Okay, ma'am.
Yeah, that's okay. No, but the notes are also being provided, right? Lecture notes. So
Yes,
try
ma'am.
to go through the notes, and if you don't
Yes.
understand any topic from the notes, you go and look that portion from the video. If it is too lengthy,
Okay.
that's why it is generally, if you each week you see go through all the videos at once, it won't be hectic at one time, right? Like for this week there were two videos, so those you can see easily, entire week is there.
Yes, ma'am.
Yeah. And for the exam purpose, so mainly I'll tell you, try to go through the questions which has been solved by the sir, or which has been given by the sir, or which we have discussed in this live interaction session. Okay. If you will be able to do those questions, that is also like make you enough prepare for the exam.
Yes, ma'am.
Yeah. Any topic you want me to add to the next live interaction session, because that will be the last one. Okay, any topic you want me to include there, you can tell me. Next time I'll come and include, or if you want any.
Okay, ma'am.
Yeah.
Okay.
About enzyme production, if you want to ask me anything, I can tell you, because that I can explain easily now. Cloning
Yes,
and
ma'am.
everything.
Ma'am, can you tell about ma'am cloning?
About cloning. Okay.
Yes, ma'am.
Okay, so very basic, let me say. So first you have to decide what is the purpose of your cloning. Okay. So accordingly you have to select a vector. Okay. If you want
Yes,
to
ma'am.
overexpress a protein in a bacteria, okay, for that you have to select one particular vector. So for the bacterial overexpression we use this vector
Yes, ma'am.
PET28A. Okay. So
Yes,
inside
ma'am.
the bacteria are very easy to grow. Experiments are very easy to do. That's why we use bacteria as a surrogate system to put our
Yes, ma'am.
gene there. Okay. In a vector
Okay, ma'am.
that is like in PET28 vector. So the specialty of this vector is like it has a T7 promoter. Okay, and T7 is a
Yes.
eukaryotic promoter. So that means all the eukaryotic genes, okay, that you want to study or to get a protein from those gene, you can put under this vector, and that will be overexpressed inside the bacteria.
Okay, ma'am.
Okay, so that is the concept behind. Other than that, if you want to perform any other experiment and you want to do cloning for that one experiment, you need to select a vector. Okay, so like
Okay.
in yeast, if you want to do any experiment in yeast, you want to overexpress any protein, so there the vector will change. There if you want to see the interaction of the two proteins, you are putting two different proteins in one vector, so there also vector will change. Okay, the selection
Yes.
marker will change. So in this PET28A vector, so there is T7 promoter, as I've said. It has multiple cloning site. It has antibiotics for the selection.
Yes,
Okay,
ma'am.
so all those things are there. So according to the multiple cloning site, you can put okay, so I'll add one more page. Okay, so suppose you want to clone one gene. Okay, if you have a gene in your mind. Okay, so in
Yes,
NCBI
ma'am.
you go, you put the name of the gene, you get the sequence from there, right?
Yes,
So
ma'am.
you'll get the whole gene sequence, starting from ATG, and then many different, and ending at TAG. Okay, the whole thing you'll get. Okay, now generally you can do many things, but main we do is the restriction endonuclease, using restriction endonuclease cloning. Okay, so
Yes, ma'am.
for that you need to design a primer first, so that you can have your gene, you can need to amplify your gene. For amplifying
Yes,
also
ma'am.
you need to have template. Okay, so suppose I need to amplify one plasmodium gene. As I am working on plasmodium, I am giving you an example of plasmodium. Okay, so
Yes, ma'am.
I have Plasmodium culture with me. Using that culture, I isolate a genomic DNA.
Yes, ma'am.
Okay, so that genomic DNA I'll use as a template. Okay, and using the database, I have got the sequence of the particular gene. Okay, that
Yes,
gene
ma'am.
is present in the genome, whole genome. So using that, I'll design the primer. So I have the sequence of this one strand of the DNA. Using that, I've
Yes, ma'am.
designed one primer, that is like one called forward primer.
Yes,
Another
ma'am.
one, with this sequence, I design one more primer. I'll take the reverse complement of this sequence. That will be my reverse primer. Okay,
Yes, ma'am.
so if you have a double strand DNA, okay,
Yes, ma'am.
so one primer will bind here, it will amplify. Another primer will bind through other strand, it will amplify. Okay, so via the PCR, you have many copy of your gene of interest that you have amplified using genomic DNA as a template.
Okay, ma'am.
Okay, so from there you can get your gene. Okay, and also, so because in the gene there will be no restriction site, right? You have to
Yes.
add the restriction site, so that thing you can add at the end of your primer. Okay,
Okay,
suppose there
ma'am.
is some sequence of the primer. Before that you can add the like suppose if you are putting BAM-H1 here, want to put, you can add BAM-H1 sequence in 5' to 3' direction, because from the 3'
Yes.
here amplification will start. Similarly, you can add the sequence here also 5' to 3'. Here if you add SAL1 enzyme, here you have added BAM-H1 enzyme. Okay,
Okay, ma'am.
so this is the extra thing that will, initially it will not bind to the first cycle. Okay, but
Yes, ma'am.
as the this strand is now formed, this new strand. Okay, so similarly, using this as a template, now many other strands will be formed. So at the end you will get the whole gene with
Yes, ma'am.
this BAMH1 and SAL1 restriction sites and the end. And now if you digest with these two enzymes, the this, yeah, this restriction sites will be generated. Okay, and similarly you have to digest your vector as well. Digest your vector. Okay, so suppose if here to I have giving two different enzymes like BAM and SAL, and suppose if you have only BAM and BAM at the both side, you have to digest your vector with the BAM only. Okay,
Yes, ma'am.
so here the sticky ends will be produced. Okay, but there are chances of self ligation of the vector. Okay, because it is
Yes.
linear, but it can again self ligate with itself. So to avoid the self ligation, you have to provide alkaline phosphatase treatment to your vector. Okay,
Okay, ma'am.
so that removes the phosphatase group. So if the phosphatase group will not be there, phosphodiester bond will not form between the two nucleotide bases. Okay, so it will
Okay,
never
ma'am.
ligate with itself. Only when you put this insert or gene of interest with this vector and provide the ligase enzyme, so only with this vector will ligate, because it has the intact phosphate group at the end of its 3' nucleotide. So these will
Okay, ma'am.
ligate. Okay, now if you have the whole construct ready, okay, now when you have the construct ready, you can put this construct into the bacterial cell. Okay, via using transformation. Okay, you
Okay,
transform these
ma'am.
bacterial gene, they will take this foreign plasmid, and then if you want to do the protein production, there are methods like you provide a primary culture, secondary culture, your cells you give IPTG, there is a chemical
Okay,
for
ma'am.
the induction. IPTG you provide to the bacterial cells. Upon IPTG, giving IPTG, these cells will start transcribing this particular protein.
Okay,
IPTG is
ma'am.
nothing but the analog of lactose. You must have heard about the lac-operon pathway.
Yes, ma'am.
Yeah, so in lac-operon
Yes.
pathway, what happens? So generally, the promoter is under the effect of this inhibitor gene or a repressor. right?
Yes.
And when lactose
Yes.
is provided, lactose will go and bind to that repressor, and now the gene transcription can be initiated. Okay.
Yes, ma'am.
So similarly, IPTG also performs the same function. It is an analog of lactose. Lactose we do not give generally because lactose will be utilized by the bacteria. Okay, so if you want to
Okay,
keep
ma'am.
your culture for 4 hours, you provide lactose. The bacteria will utilize the lactose like within 1 hour, within half an hour. Lactase will be diminished from the medium. Okay, so that's
Yes.
why you have to provide such an analog of lactose that cannot be diminished from the medium if you keep the culture for the longer time. That's why this analog is provided in place of lactose. Okay.
Okay, ma'am.
Yeah. So that is the process. You get the transcription and then protein you will get. So that's why there are different kind of this um, tissue expression cells. Okay. So these expression cells include BL21 and Rosetta. All these cells are included. Okay. They have the T7 RNA polymerase. Okay. Vector have the T7 promoter, but the T7 RNA polymerase is in the cell where you are putting it for the expression. So when both are there, then only the transcription and protein expression will be initiated. And after this whole induction process, now your bacteria has expressed the protein. Now you need to break open the cell and purify the protein. Okay, so for the purification in this vector, generally we have a tag. Okay, and again this tag we have the beads which can bind to this tag, and along with the tag your protein will also be purified from the bacterial cell extract. Okay, so that is how we perform the purification of the enzymes. So similarly there are different kind of vectors that harbor different kind of tags. Okay, yeah. So many different kinds of tags are there. With the help of those tags, we can purify the proteins. Okay. Yeah, and also if you know the property of your protein, like whether it is a positively charged protein, it is a negatively charged protein, you can apply the chromatography technique as well. Okay. If your protein is positively charged, you can apply cation exchange chromatography. If it is a negatively charged protein, you can perform anion exchange chromatography. Those you can just read it by yourself. It is very easy to understand. You can just go through the lectures. All those techniques are already taught by sir. Okay. Yes, so you want to ask anything else?
No, ma'am.
No. Okay. So should we conclude this session? Any other thing anybody want to ask? Dr. Dure, you want to ask anything? Okay, no problem. If there are no questions, should we conclude the session? Okay, let's conclude the session for today. Okay, next week again we'll meet and we'll discuss another questions similar to this for the week 12 content. Okay. It was nice interacting with you. We're concluding the session. Bye.